دنیا همش همین به اصطلاح حل کردن مسائل ریاضی و مسابقات رفتن ایناست. اینا همش خیلی خوبه. obviously mathematics خیلی کمک میکنه، به اصطلاح آدم خلاق میشه، یه جورایی ورزش مغز دیگه به هر حال. ولی بعد که اومدم توی انگلیس متوجه شدم که یک فضای غیر متعارف و یه جوری است که بازدهی خوبی نداره این کاری که ما تو ایران داریم میکنیم، به خاطر اینکه ما حتی توی دیپارتمنت متماتیکس، اونجایی که من پی اچ دی میخوندم، ما نمیفهمیدیم توی دیپارتمنت فیزیکس چه اتفاقی داره میفته، توی دیپارتمنت کیمستری چه اتفاقی داره میفته، توی دیپارتمنت بیولوژی چه اتفاقی داره میفته. در صورتی که ultimate aim اینه که ما بتونیم این چیزایی رو که یاد میگیریم به اصطلاح اپلای کنیم توی ساینس های دیگه و به اصطلاح ازشون یه چیزی پس بگیریم، بتونیم صنعت بیاریم، بتونیم چه میدونم توی پزشکی شیوه های جدید درمانی به اصطلاح خلق کنیم. بعد این یکی از ریزن ها بود بیسیکلی.
خیلی عالی، خیلی عالی. و کمک کرد این مدلینگ ریاضی در این مسئله.
آره. حالا من سعی خواهم کرد توی این جلسه امروز خیلی به اصطلاح دیپ اون قسمت‌های مدلینگ نرم، ولی تاچ خواهم کرد که به اصطلاح چجوری این یک مالتی دیسیپلینری اپروچ که تیم‌ها اکسپرت از تیم‌های مختلف به اصطلاح میشینن و با هم دیگه شیر میکنیم اون نالجمون رو و اون به اصطلاح اسکیلزامون رو. این خیلی کمک کرده. بدون اون اصلا امکان پذیر نبود این کارهایی که ما داریم میکنیم. با مثلا یه گروه ریاضی فقط امکان پذیر نبود، و یه گروه بیولوژی یا ایمونولوژیست یا متخصصان سرطان به هیچ وجه امکان پذیر نبوده. اینا چیزای خیلی خوبی هست که ما اینجا مثلا توی این دانشگاهی که من تا حالا توشون کار میکردم هست.
خیلی عالی. بسیار خوب، بسیار خوب. دستتون درد نکنه. ما که بی‌صبرانه منتظریم بشنویم این اتفاقاتی که افتاده رو شما به همون بگید و دیگه هر موقع که آقای محقق بگن و فکر کنم مناسبه. بسیار خوب. من اول از همه دوستان تشکر میکنم. لانگ ویکنده، بالاخره لطف کردن این زمان رو در وسط روز تعطیل به ما دادن. به خاطر اختلاف ساعتی که با انگلستان داشتیم متاسفانه زمان بهتری رو نمیشد ستاپ بکنیم. من کوتاه آقای دکتر هاشم کوهی رو معرفی میکنم و ابسترکت مربوط به این سخنرانی رو میخونم. بقیه جزئیات رو دیگه بذارم با خود آقای دکتر که اگر خواستن توضیحات بیشتری بدن. آقای دکتر هاشم کوهی پروفسور آف سیستم ایمونولوژی هستند at MRC Human Immunology Unit Weatherall Institute of Molecular Medicine Oxford University. ایشون همونجوری که یه خلاصه‌ای صحبت کردن دکترای ریاضیات محض دارن دانشگاه اهواز و فوق لیسانس سیستم بیولوژی از دانشگاه واریک و دکترای سیستم بیولوژی از دانشگاه واریک در انگلستان. در حال حاضر پرینسیپال اینوستیگیتور هستند در دانشگاه آکسفورد. پس از اینکه گروپ لیدر بودن در دانشگاه آکسفورد و پستاک فیلو بودن در باربارام اینستیتیو و سنگر اینستیتیو دانشگاه کمبریج. ابسترکتی که به من دادن در مورد جلسه امروز به این مضمون هست که cancer immunotherapy is defined as harnessing the power of patient immune system to find and kill cancer cells. Over the past couple of decades, cancer immunotherapy has revolutionized cancer treatment for a number of cancer types and has substantially improved patient care. Immunotherapy and/or precision medicine is obviously not limited to cancer treatment. It is in fact getting momentum in fighting autoimmune such as MS and infectious COVID-19 diseases. Despite all this benefit, it has been mainly remained unknown to the public. In this seminar, he will try to give a brief and very general overview of the new scientific achievement. He will try to illustrate what it is, how it works, and why it is important to spend millions of dollars each year on this avenue. آقای کوهی ما در خدمت شما هستیم.
خیلی متشکرم از دعوت مجددا و متشکرم از اینتروداکشن. فقط من عذرخواهی بکنم اگه امکانش هست من اصلا سویچ کنم به انگلیش، به خاطر اینکه چیزایی که من اینجا خوندم راستش هیچ وقت نمی‌خواستم فارسیشو پیدا کنم. اگه بخوام فارسی حرف بزنم یه خورده فکر کنم ناجور از آب در خواهد اومد. ولی با این حال همونطور که اول عرض کردم هر جا هر کوئسچنی بود یا هر به اصطلاح کلیر نبود لطفا منو استاپ کنید و ایدر به فارسی و انگلیسی و هر جور که میتونین سوال بپرسین، دیسکاس میکنیم این مسئله رو. ایرادی نداری؟
نه خواهش میکنم.
Okay, that's brilliant. Okay, so I think some of you may have asked yourself about this picture that I sent you in my abstract. So if you don't mind, I take a few minutes basically to describe what's actually happening here. And if you give me 1 second, I change basically this to laser point. Do you see my pointer?
Yeah, we can see.
Yes. So basically what you see here, although this is not a real image, but what you see here is this is a T cell, and this T cell is attacking this cancer cell, in basically 2 cancer cells, to kill them. And here you see that this T cell has actually managed to kill this cancer cell, and this is basically kind of what we want to do. In a sense, cancer immunotherapy is defined as how we can train our adaptive immune system, and specifically these T cells, to go through our body, find cancer cells wherever they are, and then kill them. Okay, so this is in theory as simple as this. But then the question is that why basically we call it sometimes precision medicine. The reason is that you actually never find 2 persons in the world that they will have the same T cells. Therefore, these T cells are very, very personal, and that actually makes it the question or the problem is very, very complicated. Okay, so this is very personalized. We have to take basically samples from each patient. First of all, we need to figure out how immune system work, how these T cells actually behave, how they interact with other cells, how they send signal to other cells. And by understanding that, then we can actually come up with some kind of training for T cells to tell them kill cancer cells. Okay, so that will be basically topic of my presentation today. So let me see. So for this seminar, I'll first start with a very brief introduction, and I'll tell you a few words about my career and then about my science. And as Bayesian said at the beginning, I will try to tell you what is it that we do, why we do it, how we do it, and what are our contributions, what we have done so far, and its impact in patients' health. So I actually was born in Shiraz, and in fact in a very rural area, in a village, a small village nearby Shiraz, and I did my education, primary and secondary education in Shiraz. Then I went to Kermanshah, Razi University, and did my undergrads in Razi University. And then after that, this actually was all in mathematics, pure mathematics. And actually, I graduated as a top student from Razi University. I went Tehran to Institute of Mathematics. I think at the time it was called مؤسسه ریاضی Dr. Musab, because of Professor Musab. And there I did my MS in group theory. And then I had basically a couple of years gap where I was working, and it was in Isfahan. I got married here. And then basically I moved to South, to Ahvaz University to do my PhD. And then basically the PhD program that I was participating in, it was a 4-year PhD program. And as a part of it, I could spend 9 months up to 1 year in one abroad university. And I decided to spend that time in Warwick University. And Warwick University was actually, it's very known for its mathematics. It's one of the strongest mathematics departments in the world and in UK in particular. And actually, one funny thing to note, I think again, Beshat knows more or less about that. At that time, I had 2 invitations, one from Manitoba University from Canada and the other one from Warwick University. And I was just planning to go to Manitoba University. And then what happened was 9/11 terrorist attack in New York. And then the supervisor in Manitoba University told me that, look, with the situation here, I don't think you can bring your family. And that was a big question mark for me because I couldn't actually leave my family and go Manitoba by my own. So I decided to actually change the plan and decided to go to Warwick University, and that was basically a turning point in my career. So anyway, I came here, did a 1-year program, and I finished my PhD. Went back Ahvaz University, defended it, and then I came back to Warwick University and started working there. And as Beshat said, I got another MS and another PhD from Warwick University. And then I moved to Cambridge to do a postdoctoral fellow. And then after 2 postdocs in Cambridge, one in Sanger Institute and one in Bevram Institute that I'll tell you a little bit in a second, I moved to Oxford University as a group leader. Here, group leader basically is a more independent position, more independent scientist that you can hire a number of people by yourself. You will have your PhD students, your postdocs, and then you will have your own program, basically scientific program to lead. Okay, so this is Warwick Maths Department and Warwick Systems Biology Department. This is the place I spent few years, I think 3-4 years of my career. And then, as I said, I moved to Cambridge. This is Sanger Institute. And yeah, for those who are not very familiar in this basically field, Sanger Institute is, I guess, is perhaps the biggest data center in the world. And all this, human genome first was sequenced in Sanger Institute. So that is one basically achievement in the history of this institute. I think so. It established in 2000, almost the same time as they actually sequence human genome. And when I say sequence human genome, it means that they were able to read the entire human genome sequence, and that was a big achievement. And from 2000 up to now, so far, they have actually had a couple, if not more, Nobel Prizes. And then I moved to the Bayesian Institute, again in Cambridge. I spent my second postdoc, nearly 4 years in this institute. And then in 2017, I moved to Cambridge, moved to Oxford University. And this is basically a research institute. It's called Wetherall Institute of Molecular Medicine. This is a research located in John Radcliffe Hospital in Oxford. It's the biggest actually hospital in city of Oxford. And again, I think it's established 1990s or 2000, but they have contributed quite a lot into human health and medicine. And a couple of years ago, they had one Nobel Prize, actually, office. The scientist was sitting here, and this is basically my office here. Okay, and then, yeah, then I was in Iran when I was doing my PhD in Ahvaz University. Professor Karamzadeh was my supervisor. Professor Motamedi was my advisory member of advisory board, and Professor Haqqani from Technical University of Isfahan was my examiner. And then later on, basically, I made family friendship with all of them, and I visit them whenever I get chance. Unfortunately, Professor Motamedi passed away a few years ago, but Professor Karamzadeh and Haqqani now is in America. I'm in contact with them. And then in Warwick University, I was working with this very renowned professor, Hajar Nevis, is a very well-known mathematician. Still, actually, I have family friendship with them. I go there. They are old and retired, but every now and then I get chance. I give them a visit. This is last time, a few years ago, they visited us with his son, and we were basically, it was summer, and we were eating ice cream. And then in Warwick Systems Biology, these two incredible people actually made quite a lot of impact on my career. Professor David Rand, who was the director of maths department when I joined the department, actually he helped me quite a lot, because I think one question at the beginning was that what happened that I switched from maths to biology was basically mainly because of Professor David Rand and Professor Alison Roger. And one big help that he made, basically, with my PhD that I had from Iran, I applied for a home office work permit, because we had to take basically work permit to be able to work here. And they kind of rejected my application because I didn't have my original certificate. What I had, it was just a copy of my PhD certificate. And then what actually Professor Rand suggested was that, okay, what if we just register you as a student, then we will not have to follow home office rules, because we don't need work permit for a PhD student. And if you finish this job, then Warwick University will issue you a second PhD. And basically that was a very good idea, and I followed that, and that was a big turning point in my career. And the same Professor Alison Roger, that at the time was the director of systems biology again, or co-director, she helped me quite a lot, and now she has moved to Australia. Still, I'm in contact with both. So why is not moving? Okay. So is there any question up to here?
I don't see anything in the chat, so you go ahead.
Yeah, okay. Now, basically, after that introduction about myself, I am going to tell you a little bit about my research. Again, that was a question at the beginning, that what we do and whether basically maths is of any use in what we do. And the answer is absolutely, because basically my research interest actually is actually sitting here at the intersection of immune system, how immune system works, and mathematical or statistical inference or modeling, and also machine learning and artificial intelligence. So in a sense, in basically each project that we work, we have some expert from immunology, some maths or statistics, and machine learning and artificial intelligence and data science. And the key questions at the very top level, as I said earlier, is that we need to know how T cells are developed at the beginning, and how they function, how they interact with other cells. And if we fully understand these things, then we can come up with some new ways to basically train them, to modify them, to follow our instructions. Sorry, sometimes this starts moving. Okay, so just a little bit about immune system for those who might not be very familiar with. Our immune system has basically two arms, one innate arm and one adaptive arm. Innate is basically, a very quick reaction to a disease. Those cells, basically, these are a population of cells that actually quickly react to a disease for from the beginning, from the time that actually you face a disease up to a few days. And then after that, adaptive immune system get involved. And the reason they call it adaptive immune system is because basically they get rid of the disease, whatever it is, either cancer or infectious disease. And then basically they leave a memory. And that memory stays in our body for long, long time. And that is basically foundation of vaccination. And in fact, this immunotherapy that I referred at the beginning, his stories, I'll actually tell you a little bit of about history of that. Although the terminology is new, what we have been doing is not actually very new, because if we think about vaccination, it goes back nearly a century or even more, because we have had vaccination for a long, long time. Anyway, so the good thing about this adaptive immune system is that they leave a memory, and that memory is crucial for protecting us against reoccurrence of the disease. And one point to note is that the adaptive immune system has basically two type of immune cells. One are called T cells and B cells. So these three are subpopulations of T cells, and this is population of B cells. There are some other basically that are sitting at the intersection of innate immunity and adaptive immunity that today I don't want to talk about this. So the key point here is that we are working on adaptive immune system. You will see why. And adaptive immune system, basically two key pillars of it. One is B cells and one is T cells. So if we look into basically a timeline of actions or mechanism of action for B cells and T cells, this is what we get. So basically here, what has been illustrated in this x-axis, we see basically time in days. And in y-axis, it's just a cartoon and illustrative of immune response. And what you see here, this is a cancer cell, basically, or an infected cell. It doesn't really matter because, in reality, the action is the same from adaptive immune system point of view. So what happens is that a T cell come across with this cancer cell, and then as soon as it recognizes this cancer cell, it's activated, and then it actually inform other B cells, and they quickly give rise to an army of T cells and B cells, and you see this immune response goes up, basically. This is the peak of the immune response. And this is the time that basically your adaptive immune system manages to basically win the problem, whatever it is. And then after that, when they are in control of the disease, because the body doesn't need to all those army of B cells and T cells, most of them die out, because if the body wants to keep them, it will come with the cost of, they need energy, they need nutrition. It's really, really difficult for body. And therefore, most of them, they die out. But a few population of them that are called either memory T cells or memory B cells, they actually live in our body forever. And important thing here is that if you look at the y-axis, the immune response started from 0, but after disease, the immune response doesn't go to 0. And that actually stays in our body for long time. And that is another point behind vaccination, okay, or behind personalized cancer immunotherapy that I will tell you a little bit more later. So now, by knowing that is basically B cells and T cells are basically two key pillars of adaptive immune system, our task is to find out how they work, how they basically communicate with other cells, and how they find, say, cancer cells, and how they kill them. Okay, so one question you might ask, or you might have, or wondered, was that if really these T cells or B cells kill cancer cells, then why the disease, the cancer, is very deadly, and we see all people are dying. And the reason is that basically it's very interesting phenomena behind the scene. I'll try to basically go back how cancer basically is developed. Hopefully that will give you a hint of what is happening. But to put it very simple, is that after up to a period, your T cells or B cells kill cancer cells and nobody knows. For instance, perhaps in my body, now there are some, so what we call it benign cells. These are trying to give rise to cancer, okay? So perhaps in my body I have, and actually very certainly I have, each person will have, but because my immune system, they kill them, that's okay, we don't actually see that. But the problem is that at some point of time, these cancer cells start developing some way of fooling T cells and B cells, and therefore T cells start believing that that cancer cell is a normal cell, and they stop killing them. And that is the problem. And that is the reason then, cancer take control of the body, and then the patient will end up either in hospital, and shortly later they will die. Okay, so that's the issue here. So then, to just summarize, basically our focus or our basically aim is to develop or to find ways of training adaptive immune system to find and kill cancer cells, and this is known as cancer immunotherapy. And one point to note here is that I just started by cancer immunotherapy because it was easier to illustrate, but the same principles apply to infectious diseases, or slightly differently to autoimmune diseases, and so on and so forth. So at the behind the scene, the same principles are there. Okay, and the advantages of basically cancer immunotherapy is that they are very precise, in theory at least. They actually kill tumors, not healthy cells, and that is basically very, very important. They are systematic. They can find and kill cancer cells anywhere in the body. And again, another basically point to note here is that usually, you have here, we say, for instance, breast cancer or lung cancer or skin cancer, okay? The reason actually we hear this, because this is the tissue that gives rise to cancer. But at this stage, the cancer is called basically primary cancer. And in medical term, usually they are actually labeled as, I think, stage 1 or stage 2. Okay, and then after that, cancer cells actually move to all over your body, and they go everywhere basically. And that is the reason surgery is not an option anymore. So if you have a cancer that has actually moved everywhere in your body, or in a medical term, it's metastatic, it's basically the surgery or other treatments are basically out of option. They do not work. And then it's very adaptable. And the reason is that, as I said earlier, tumors evolve all the time. They come up with new ways of basically fooling immune system or escaping immune system. And therefore, if we actually develop a good immunotherapy, then they are very adaptable, meaning that they will evolve alongside of cancer evolution. And they are very universal, meaning that they can be applied virtually to all cancer types. And they are durable because, as I said earlier, they give rise to memory cells, and these memory cells actually will stay for a long period. So it looks like that there is a question, yes?
Yes, there is one. Actually, Payman says, is this model generalized for all type of cancers? Also, you are talking about vaccine for cancer. Could you please explain more if we have any type of vaccine for any type of cancer?
Yeah, that is a brilliant question. Basically, both actually. And I was actually going to tell you about a bit later. The first question, that whether this actually works to all type of cancer, the answer is at the same time yes and no. Yes, because basically now we know that which cancer types are better. Basically, this immunotherapy is better approach for that. And one thing to note that I'll explain a little bit later. So far, when I say cancer immunotherapy, I refer, this is a very general term, I refer to different types of basically training immune system. And the reason that we have different types of immunotherapies, one reason because each immunotherapy works for one cancer type better. For example, if we have, say, a skin cancer, you will see later that what we call it is called basically immune checkpoint treatment, that I will tell you a few minutes later. For skin cancer, work very well. Also for lung cancer, it works well. But when it comes to blood cancers, this particular treatment doesn't work. We need to actually switch to a different type of cancer immunotherapy that I'll explain a bit later. And then the what was the second part
vaccine
of the
question?
Yeah,
is there a vaccine?
Yeah. Again, that is a brilliant question, because one of these basically cancer immunotherapy is what we try to do is to develop personalized vaccine for cancers. And in fact, the person who basically developed a Pfizer vaccine, who was originally a cancer scientist, and in his basically both his wife and himself, they were working on cancer, and they had developed personalized cancer immunotherapy at the trial level. And upon basically pandemic, they switched from cancer, and they say they basically use the same technology to come up with this cancer vaccination. So, sorry, the COVID vaccination. So the answer is yes, we develop, and you will see a bit later. I'll actually explain how it works.
Thank you, Asham. But Pema also asked, based on your explanation, it seems that there won't be any chance for the occurrence of the cancer cell in the body. Is it right?
Well, it depends. And again, it's a brilliant question. I don't know what's the background for Payman, but he's actually asking brilliant questions. Basically, you see all these things I have put here. And at the beginning, I said at least in theory, we already have developed these immunotherapies. I mean, when I say we, it's not me, all the experts around the world. Yeah. And if that is the case, you might think that, okay, the question has been solved, and what the hell are you doing? But the problem is that the same way that immune system is very variable, and we don't see each two person that have, the same immune system, also cancers are very different. When we say that a person has cancer, say lung cancer, with another person with this lung cancer, from our eyes, these two are not the same thing. We need to basically look at, to the details of it, and see what's going on, what type of, genotypes they have, and so on and so forth. And therefore, to answer the question is that, unfortunately, the response that we see from these cancer immunotherapies are very variable. And one basically focus in my research group is that to find out why this response is very variable, why some patients respond to this immunotherapy very well, some other patients, they do not respond, some other patients, after that, they develop some side effects. And actually, I was going to explain later, but perhaps it's a good time because there was a question here. What we do, in a sense, we train or boost the power of T cells to find and kill cancer cells, okay? But the one complexity here that if these T cells are not trained well, and if we push a bit, too much, then they will start killing normal tissue and normal cells. And then what will happen would be that the patient will die not because of cancer, but because T cells are killing their healthy cells. Okay, so it's basically very complicated. And if we have some patients, we have some examples that the memory cells have actually stayed there, and after say 30 years, the patient is still alive, and they live their lives.
Okay, thanks, Hashim. Quick question from myself. When you say personalized, what do you mean? Do you need like blood sample, or how do you personalize that?
Yeah, that is also a good question. Basically, it's kind of related to that vaccine question that I said. Basically, we developed a personalized vaccine. Again, you will see a little bit more. Hopefully, it will be clear by end of this seminar. But one way of cancer immunotherapy is that when we basically when we come across with a cancer patient, we take 2 biopsies, one from tumor and one from healthy tissue. And by comparing these two via some mathematical modeling and algorithms, then we find out the signature of that cancer. Okay, and that is the reason I said that if you have basically 2 persons both having lung cancer, it doesn't mean from our eyes it's the same, because we need to find out what actually went wrong in the patient A genome that gave rise to cancer in patient A, and what went wrong in patient B's basically body or genome that gave rise to cancer in patient B. And based on that, we need to basically train the T cells in a way to basically work only for that person. And that is the reason it's called personalized cancer immunotherapy.
Good, thanks. We have a question from Changiz. He says, is it possible to develop T cells out of the body in the lab and then inject them to the patient to boost their immune systems?
Yeah, basically you are just telling me, what I have in my next exercise. You will see that. That is another type of cancer immunotherapy. Perfect. Yeah. Good. Okay. Yeah. Good. Okay. So can I carry on or?
Yes, go ahead. We don't have, well, there is one. Oh, no, we don't have any question. Please go ahead.
Okay. So basically, another point to make here with this background that I have actually told you, hopefully by now it's slightly, more clearer that for us, on one hand, we need to basically understand T cells. And on the other hand, we need to understand tumors. Okay, and the interaction between these two is crucial. And that is very specific, basically, research focus in my group, how these T cells interact either with cancer cells or with infected cells. Okay, and by doing that, obviously, it has application in cancer and cancer immunotherapy, both in understanding how cancer is developed and it's actually evolved, and how to develop some sort of cancer immunotherapy. And also the same principle applies for infectious diseases. I will show you some of our work, in finding targets for COVID-19 vaccines. and also similar principles are applied to autoimmunity and autoinflammatory diseases, such as inflammatory bowel diseases. Again, we have actually contributed to this field quite a lot, and at the heart of all of them is to understand how T cells interact with these target cells, whatever it is. So now let me actually give you a little bit of a history of cancer treatment. What basically has been recorded is that the first surgery that actually took place in the United Kingdom, it goes back to 1800. And then it took nearly 1 century to develop another basically turning point in cancer treatment, and that was radiotherapy in 1900. And then half a century after that, we got chemotherapy that is actually very commonly used, still is one of the very common treatments that for all cancer types are actually used, pretty much all. And then from the big, and I have actually color-coded this in red and green for to make one point. And the point is that up to here, up to the beginning of 21st century, the treatments were mainly targeting cancer cells. So in surgery, you take basically the tumor out. In radiotherapy, basically you with radiation, you target cancer cells and kill them. With chemotherapy, basically because these cancer cells, they give rise to tumor. They just divide and divide and divide. Basically, figure out which cells are dividing and kill whatever it is. All the cells, no matter cancer or non-cancer, and by that, obviously, you are killing cancer cells. But after that, it comes back. So, in a sense, up to here, the treatments were mainly targeting cancer cells. But at the beginning of 21st century, scientists noticed that actually, if we can target immune cells and train them, they will actually go and find cancer cells and start killing cancer cells themselves. And that is basically, that was the turning point. At the beginning, we had these targeted drugs. And then in 2010, this Journal of Science, which is actually a very influential and highly reputed science magazine, it's called the year as year of breakthroughs in cancer immunotherapy. And that was actually very, very important. And that happened in basically, the terminology started to appear in literature in 2010, and then science basically labeled breakthrough in cancer immunotherapy in 2013. And in 2016, basically in United States, we saw basically this person here is Sean Parker, and Sean Parker was the co-CEO of Facebook, basically. He decided overnight to put $250 million to develop an institute called Parker Institute for Cancer Immunotherapy in United States with a new basically way of research. And funny enough, in that night or in the opening of that institute, he invited not only very highly reputed scientists, but also celebrities around the world. For example, Nicole Kidman was there, Tom Cruise was there, George Clooney was there, and quite a lot more. And I think his intention was that to bring public attention to this turning point in the history, to tell them that, yes, actually, there is an opening window to cancer treatment, and that actually gives rise to hope that we can actually eventually cure cancer. And that was the beginning. Now it has actually billions of dollars in market, worth of billions of dollars in the market. And then what happened in 2019? Then these two scientists, James Allison and from United States and Tasuku Honjo from Japan, they got Nobel Prize for one type of cancer immunotherapy that they had developed that I'll tell you a little bit later. And since then, actually, there has been numerous different cancer immunotherapies, big achievements that I don't have time to go through it. Okay, and I can actually stop here if there is any question, because then I'll give you just a few stats about how deadly cancer it is and why it is forced to spend billions of dollars treating them.
I don't have any question in the chat. Is there any question in the room that somebody wants to ask? I don't hear anything, so I think
Okay.
yeah, you can go ahead, please.
Okay, so basically, if we just look at numbers, what we see is that new cases, actually these numbers that I've been looking at here is up to 2018. Each year we see nearly 17 million basically, and the deaths are nearly 10 million. And interesting enough, nearly a third of them are associated to smoking. So smoking is actually a big factor. And 16.5 million people will die from cancer in 2035. 40% of people will get cancer in their lifetimes, unfortunately. And $151 billion, the cost of cancer in US alone in 2018. So you see that this is a massive burden, both for healthcare, for social and family lives, and also to the economy. And therefore, this new, basically this cancer immunotherapy brings hopes to perhaps, makes this happen, makes, cancer treatable. And if we just quickly look into some numbers of what cancer types are more common in both new cases and also death, is slightly different, obviously, for men and women. For men, we have the highest new cases is prostate cancer, and 20% of basically the whole cancer diagnosed cancer patients are diagnosed with prostate cancer. And then we have lung cancer, colon cancer, and so on. And not surprisingly for ladies, the top is breast cancer, and then lung cancer, and so on. And then when we look at the death, basically numbers, what we see basically lung cancer, which is highly associated with smoking, is the most deadliest cancer, basically. And then we have, some other cancer types, and this is also I've highlighted to basically just make the point that this is also associated with diet. So the point I want to make here is that you will see later that cancer basically is initiated upon mutation in the genome, and that mutation basically happens random, and it actually happens all the time, because each cell has to divide, and upon this division, the DNA is copied to 2 DNAs, and something goes wrong. But there are some other factors that actually enhances the possibility or the chance of developing cancer. So if we look at, again, some numbers over years from 1975 to 2016, here are the new cases and here are the deaths. So what we see is this is for male, for female, and combined in the middle. Okay, and basically there are a couple of points to make here. One is that you see up to almost beginning of the 21st century, this trend was actually going up, and then started actually or changed the direction at the beginning of 21st century. And this is very likely due to development of one for this cancer immunotherapies that actually prevents the death, and also because of the awareness and associating cancer with some factors and asking people to if they can avoid those factors. And another way to highlight the importance of this cancer immunotherapies is in this figure for men and women. Basically, if we didn't have development of this cancer immunotherapies, then this is what you would actually see. The rate rate of deaths in men over the these years that have been highlighted here. And this is what you would see for the women. But what I have actually colored differently here is that in reality, this is what we see basically. And the reason that basically we have kind of broken this curve is again partly due to development of these new technologies and new ways of treating cancer or cancer immunotherapy. So the point I think I'm going to make, at least one point, is that those people or those governments that they spend this much of money, they really know what they do. Or Sean Parker, who decided to put $250 million overnight from his pocket to develop a cancer research institute. He knows what he does, and he knows basically the impact of this in the future. Okay, if there is any question, I'm happy to take. Otherwise, I'm now going to take one step back and tell you how cancer starts, basically.
Well, I don't see any question in the chat. Is there a question in the room? Or if there is, please unmute yourself and ask your question. No, I don't see anything.
Okay.
You can go ahead.
Yeah, I'm just a little bit worried that if sometimes I don't explain it clearly. Just I need to know so that by end all the points are made and we know what's happening and what we do. Hopefully.
Sure. Thanks.
Yeah. Okay. Now let's have a look to slightly in more details how cancer is developed and what we do. So if I take one cell by random from a patient and open that cell, obviously the cell has different compartments. The most important perhaps is this nucleus. And in the nucleus, we have our DNA, or the code of basically life. And for humans, each DNA is basically built of 23 chromosomes, and this is the shape of one chromosome. And then if we start opening up basically this chromosome, we see that DNA is wrapped around this small molecular proteins. These are called histones. And even if we further open up DNA sequence in a linear manner, this is what we see. And the important point to make is that the DNA, which is basically, again, the code of our life, is actually written in these 4 letters, or with these 4 chemicals: guanine, cytosine, adenine, and thymine. And here, for the simplicity, we will just call them G, C, A, and T. Okay, and it's actually double helix. As you see, these double helix are actually stuck to each other. And a few simple facts is that obviously each cell carries this DNA molecule. And if we just open it up and measure that, the DNA molecule is 12-meter long. And interesting enough, this 12-meter long DNA is, very nicely and very geometrically basically compacted into a very tiny molecule that sits in the nucleus. Another fact to know is that if we want to basically print the code of life, just G, C, A, and T, it's not the whole chemical element's name, it will actually fill 200 books, each 500 pages. And actually, in one of the universities that I was working, the chromosome 23, which is I think it was chromosome 24, or yeah, it's the smallest chromosome. They had printed part of it in a wall, and it's the whole thing, I think, is printed in and is kept in one university. I think it's in Leicester University. So anyway, the point here, one point is that this is so crucial. Cells know what to do because the instruction is written in this DNA code. And we need to understand that. And basically, the first time that human managed to read this whole genome was year 2000, as I said earlier, in Sanger Institute, in collaboration with a number of institutions in America. Okay, now let's have a look to how basically this works. So we have this DNA, as I said, it's double helix and it's written in this 4-letter alphabet. And then DNA has to be first transcribed into RNA, okay? And the reason, the simple reason for this is that because, RNA is very similar to DNA. All basically the difference, the only difference that you see is that this T is actually changed to this U, but this is not a big difference. But the reason basically cell does this is because this DNA, the cell has only one copy of this, and this is very important, and they need to actually keep this molecule safe, and therefore they don't want to damage it, and therefore basically there is a copy made from this DNA. That copy is called mRNA. And then this mRNA or RNA in general is translated to a protein sequence. And the way it's translated is that it should be in codons, basically. Each 3 letters are translated into 1 amino acid. So you say this 3 first letter translated to 1 amino acid, I'm sorry. The second codon is translated to another amino acid, and so on and so forth. So in a sense, what I'm going to basically say is that you have DNA that gives rise to proteins, and these proteins are basically the key molecules that actually keeps our body running. Okay, basically in a healthy cell, this is what happens, and this simple principle is called the central dogma of cellular biology. But then what happens that actually it gives rise to tumor or cancer? The thing is that basically cells divide all the time. Obviously first you have an egg and a sperm, they actually combine one cell and then they start giving rise to different cells. This is one reason for cell division. Another reason is that some cells actually die because their life is over, and therefore you have to have new cells, and therefore in your body all the time cells divide into different new cells or sister cells basically. And in each division, basically what happens is that the DNA has to be copied. You have one DNA, that DNA has to be copied into 2 cells. And again, here I have illustrated what happens. Basically, we have this DNA, and then sometimes when a cell basically makes 2 copies from the DNA, something goes wrong. For instance, here you see that it was T, and that actually changed to G. And sometimes it's not just a single nucleotide sitting here. You see that basically 3 extra nucleotides inserted here. And these things happen by random. Or sometimes one actually is deleted. Okay, it's better illustrated here. So this is a normal cell. This is the DNA. And this DNA in a normal cell gives rise to this sequence of proteins or amino acids. But what happens is that so the first basically type of mutation is that this C here randomly just changed to J, and because that changed to G, then this CCT was giving rise to this amino acid, but this is translated into different amino acid. And then what happens is that because of this amino acid, this protein is changed, and because this protein is changed, the function of cell changes. And therefore, for instance, a cell that has a lifetime, when its lifetime is over, it doesn't die, basically. That is one reason. And we have different types of mutations. So here it was a substitution. Here is an insertion. You see that this DNA is 1 nucleotide longer because we had 1 insertion here. sometimes one is deleted, and so on and so forth. And most of the time, we are lucky, and these basically changes are harmless. They don't cause to cancer. But unfortunately, that is not all the case, all the time, the case. Sometimes they actually give rise to cancer. This is one fact. And another fact is that it doesn't mean that as soon as, a mutation happens and it gives rise to a benign cell that is actually primed to give rise to cancer cell, it will be a cancer cell, because as I said at the beginning, up to a period of time, our immune cells, they find them and they kill them. And I'll tell you in a second how immune cells actually find them and kill them. But at some point, these cancer cells, they basically develop a new ways to basically fool T cells. So in a sense, we had this tumor here, healthy cells. In that healthy cells, one cell actually encountered a tumor, a mutation. That mutation in the next division gives rise to two. Perhaps that actually another mutation encountered here. And eventually you will have this tumor. These are cells disobedient. They don't die. And they eat your energy. And they actually, first they start from a tissue such as skin, such as lung or blood or bone or things like that. But then once you have this tumor developed, they actually go to other tissues all over your body. And basically the disease, I was telling a while ago to my son, it's not something very, unusual. The reason actually that actually kills you is that because they eat all of your energy, all of your body's nutrition. And then that is the reason you see that cancer patients at the end of their life, they don't have even energy to go from one room to another room, basically. That is one reason, obviously, not the whole reason. Okay, so this is how cancer is basically initiated, but then how cancer cells inform other cells, and in particular T cells, to tell them, hey, there is a problem here. I'm actually having a problem. Okay, now let's again open up this cell. What we see in the nucleus is this DNA. So in this example here, I'm talking about cancer, and because of that, it starts with a mutation, and here it has been highlighted in red. Okay, that this DNA, as I said, is transcribed and translated and gives rise to this protein sequence. Each of these small spheres is actually illustrative of one amino acid. So a protein sequence is basically a string of amino acids. And this amino acid here was the one that is unusual because basically it appeared here because of the tumor, because of the mutation. It's not basically a normal or healthy amino acid sitting here. Okay, and just another point to make here, if you think about infectious diseases or viruses, again, it's the same. So what happens is that virus enters your cell, and then actually their DNA enters to the nucleus, and then the same principle, that DNA is translated to protein, and then the same pathway up to this alter explain. So everything is the same. Just I use this cancer because it's easier. Therefore, so what's happened here? We have this protein, long sequence. In the cell, it's actually transported to another compartment called proteasome. There it's chopped into small sequences. It's called peptides. These peptides, so here I have illustrated 3 of them, but you can think of millions of them, okay, in the cell. They actually go to another cellular compartment called endoplasmic reticulum. And there they actually are loaded to these HLA molecules. These are another proteins. And similar to blood, that people have different blood types, people have different HLAs. And actually, the HLA types is more variable than blood types. And it's very important, and they actually play an important role in immune system. And actually, you see that sometimes a patient needs, say, a kidney transplantation or whatever tissue transplantation, and they say that we need to find a match because those two patients should have the same HLA. If they don't have same HLA, then as soon as you take a kidney from a person with different HLA, put into a patient from another HLA, then quickly adaptive immune system, they will think that this is basically a threat, this is a foreign object or foreign cell. They will start killing them, and then what we call this tissue rejection. Okay, so that is the reason. So back to the basically cancer story, these small peptides that usually have something between 9 to 20 amino acids are loaded to HLA. This HLA, not all of them, some of them, they come and sit in the cell surface. In a sense, the way you can think of, this is a barcode of cancer sitting in cell surface in the form of this HLA. And again, the variability of this HLA, otherwise cancer, immune cells could actually kill normal tissues. Okay, so that is the key point here. So then this is the way that a cancer cell or an infected cell tells the outside world that there is something wrong inside me, please help. Okay, so that is a key point. And basically, one thing we do here that it was asked, a couple of very interesting questions, that whether we develop personalized vaccines. The answer is yes. And what we do, we take a patient and we try to predict with mathematical modeling what basically this peptide will sit here that it will actually activate T cells. And then we use this as vaccines. And by putting that, then T cells know that whatever cell that has this barcode is a cancerous cell, and they will kill them. And the reason is that this is very personal, is because two persons have different DNAs, and therefore these mutations are different in different people, and therefore this that we predict is basically very personalized. Okay. Any question?
Yeah, I have a question. For how long that would be effective? Like with that vaccine that you said, you inject to that person, personalized vaccine, how long that would be effective?
Yeah, that is a good question. It's very variable. We have examples. Actually, first I had put a story here, but then because of time, I removed, and it's about a scientist that in age 31, he became the director of Cancer Research Institute in America. And that was the first time that basically one scientist at this age, because it's a very prestigious job, became there. He was cancer scientist, and he was developing different types of basically cancer immunotherapy. And then he came across with a lady, a cancer patient. I think at the time she was 19 years old, or even not 19 years old, a member of US Navy. And because she had spent quite a lot in sun, very likely that was a factor that she had developed that very severe skin cancer. And other doctors had told her that you don't have more than 4 months time, and they had developed pretty much whatever they could. But Steve Rosenberg had actually come up with a new way of cancer treatment, but he hadn't basically tried it. He hadn't experimented it. Because the lady was going to die anyway, they decided to go ahead and try that with this lady. And basically they started, it didn't work. Other scientists came back to Steve, they told him that it's not working. He asked them to increase the dose. They increased the dose at the level that all other scientists were fed up, and they came back to Steve that it's not working and you're killing her. He had 4 months, that you're basically not giving her 4 months time. And his response was that this is not the life she has. basically, but I'm almost sure that this should work. And actually, some people believe that he had prepared his resignation letter. But after, I think, 10 days, they started to see the sign of improvement. And after 2 weeks, the lady came out from, ICU. And then after a month, she became cancer-free. And then went back to her normal life. And then 30 years later, they actually managed an event for Steve, for that lady to meet Steve. And it was a very emotional moment. So we have patients like that. And actually, I got to tell you that at the time that she freed from hospital cancer free, basically the stock value for that treatment went up millions of dollars in US in one night, basically.
So during the 30 years, there was no revaccination?
No cancer, but it doesn't mean that, we have now a treatment that it will work for all skin patients. The problem is that the response is very variable. For cancer, for some patients it works, for some not. I will give you a figure that it actually tells you in average how it works. Okay, but
before we go there, Hashim, there is a question. Hamid wants to know about your mathematical model, because a couple of times you mentioned that based on the mathematical models, we do this, we do that. So is there something that you can share with us?
Yeah. I have some actually later, but one, obviously I haven't put the equations or, what we do here, but there were actually a couple of points I made here. Okay, one was that here at this cellular compartment, there are basically millions of these peptides here. Okay, and then some of them are actually managed to be loaded to this HLA molecule and sit there. So the very first question from mathematical point of view is that how you can develop a model to basically take this DNA sequence and predict which of these peptides will actually sit in the cell surface. Okay, that is, okay, this is very, mathematically oriented question, and it's quite a lot of maths and programming and machine learning behind that.
Sure. Hamid wants to specify something. Hamid, please unmute and tell us what you want to know.
Thank you, Rashad. Hi, Dr. Kui. Thank you so much and for your presentation. My question is about the type of model that you used. Have you used the previous data that you had to train a kind of like neural network or some artificial intelligence to predict the things, or you just because you said that you try to refine the data, have you used any statistical method? Just I just want to know, elaborate that part. Is it the
absolutely, yeah.
Sure. Thanks, Hamid.
Yeah. Okay. So there are as I said, this is a very multi-step and stochastic process, and one highlighted here, and this one behind basically this is mainly deep neural networks. But these questions or these models are more mature. I mean, what we have now out there are some models that actually can predict which peptides loaded to HLAs will be displayed here. But a more difficult question that we work in our group specifically is that from those that are displayed in the cell surface, which ones are what we call this immunogenic, meaning that which ones will actually activate T cells. Again, this is not the case that all those that sit here, they are actually activating T cells. And then here we take a combination of different approaches, and that is all the time we basically do to see which one it will work. So we have developed some Bayesian models. And for that, obviously, we have quite a lot of data from those that actually trigger T cell response. In a sense, those are immunogenic. And then we have a list of them that they, in the lab, they were actually, evaluated and they didn't trigger any T-cell response, non-immunogenic or negative. And therefore, again, considering different HLAs, we can take hierarchical, Bayesian approaches to shed some lights on which one are better targets for vaccine. And actually, one of the publications that I will show you later was that at the beginning of the pandemic, we actually used this strategy to quickly predict which part of the SARS-CoV-2 virus are more likely to sit here and trigger T-cell response, meaning that these parts or these small peptides or epitopes are those that they have to be used in vaccine development. And there will be some more hints for mathematical modeling a bit later.
Thanks. There is a question, Hashim, that says, based on the previous slide, it seems there is a compound probability pre-mutation for generating different type of DNAs. So how to model all of them by considering all of the probabilities?
Sorry, read it again, please.
Well, it says, based on the previous slide, it seems there is a compound probability permutation for generating different type of DNAs. So how to model all of them by considering all of the probabilities?
Yeah, I'm not sure I understand the question.
I think what he meant is, you said that's very random that this mutation happens, changing in DNA from G to T, T to G, something like that, amino acids. So his question is, so there are so many different probabilities out there. How you can model that considering all of those probabilities?
Okay, so I think at some point, basically, I said that to start with, we take 2 biopsies, one from tumor and one from healthy tissue. And then what basically our wet lab collaborators or biologists do basically take these samples, they extract the DNA, they send to some other laboratories, they sequence them, and then what we get is the DNA sequence of this. Okay, and again, at the beginning or earlier, I said that genome is massive. I mean, if you just want to measure it, it's a massive molecule. But the thing is that basically only 2% of the genome gives rise to proteins. In a sense, 2% of genome is protein coding area. Then that 98% is basically functional part of the genome. And for cancer, and for the sake of simplicity, so far, basically what I'm talking about and what I'm showing you here is that 2% of the genome. Okay, and therefore, by comparing healthy tissue versus basically a tumor, first we can actually identify all the mutations, and then some of these mutations are basically harmless. We can actually compare this if you see a mutation that exists in tumor and also exists in healthy tissue, then perhaps that is not something very cancerous. And then what we are left is a bag of so-called somatic mutations, and those somatic mutations are perhaps those that are more likely to give rise to tumor. And then we study all of them, and by use of this deep neural network and other machine learning models. Again, here I illustrated only one example. Obviously, for machine learning models, it's easy to investigate all the mutation that we get from each cancer patient.
Okay, thanks. So we don't have any other question at this moment, Hashim. Thanks.
Okay. Okay, and then, so up to here, what I described was that how a cancer cell or an infected cell tells to outside world that there is a problem inside. And now another basically key component of this story is how immune cells actually developed, in particular T cells, and how they recognize either cancer cells or infected cells. For that, let me put one step back and look at B cells and T cells. These cells, both B cells and T cells, they have one characteristic which is different from other cell types, and that is their receptors. In their surface, basically what we see for B cells is this shape here, and for T cells is slightly different. But important point here is that this receptor has basically two chains. It's heavy chain and it's light chain. And each chain has a variable region and a constant region. Okay. And then basically this variable region, that again, you will see more about that a little bit later, is the part that basically come to contact with basically these peptides here. Okay, and that is perhaps I would have to basically explain this with T cells because our focus is mainly on T cells. So this variable region at the end of the day is this part that come across with this peptide and scans this and basically understand if this is a healthy cell or is a tumor cell. Okay. But how these variable regions are defined, and another basically hint to what are the contributions of mathematical modeling. So let's have a look here. So here I'm going to tell you how this part here are defined. Okay, if we look at in our genome, actually this is from our previous publication that we were looking into a mouse genome. So to be precise, this is a mouse genome, but not human genome, but they actually work very similarly. What we have 195 so-called variable genes. These are gene segments. These are part of your genome, basically, okay? Their length is different, although here I have put basically the same, rectangle, the same size rectangle, but there's some of them are very big, some of them are small. And then slightly down or farther away, you have 10 diversity genes, or D, and then farther away, you have 4 J genes. And what happens is that first, a D gene is randomly selected, a J gene is randomly selected, they combine with each other, they give rise to this DJ segment. Okay, and then this DJ segment is actually combined with another randomly selected from this pool of 195 to give rise to this VDJ. And you might ask that, okay, what is the purpose of this random selection? This is the way our immune system increases the diversification. It wants to basically make it as complex as possible so that viruses and pathogens do not understand what is the code, what is the immune code. Otherwise, if it wasn't the case, then viruses could actually easily wipe human population out of the world, basically, okay? And that's not all the complexity. Another layer of complexity here that I haven't shown here is that this is not exactly, that these gene segments are, combined with each other. In middle of them, so between D and J, some nucleotides are randomly inserted or randomly deleted. And again, the reason is that to increase the diversity. Okay, now our task is that at the end of the day, we need to figure out from this very random, basically, system, what T cells or what B cells the patient has. Okay, and here I'm talking about basically 10 to the power of the possibility of this, considering everything. I'm talking about some estimation is 10 to the power of 60 or 10 to the power of 50. It's massive. It's more than all the stars in the universe. And for that reason, if we take blood sample from 2 patients, we don't see similar T cells. We very, very rarely see similar T cells from the same patient. And in particular, the patient is very basically healthy. If a patient, say, they have been exposed to a virus, then perhaps a T cell basically recognized the virus, activated, proliferated, and it gives rise to massive population of T cells, and those T cells are very, very specific to the virus, and therefore that T cell is populating your blood. And for that situation, yes, we see a few T cells from patients A is similar to patient B. Otherwise, if you have 2 healthy patients, it's really rare to get same T cells from 2 different individuals. And what I showed you here was from 1 allele, because usually we have 2 alleles, one from mom, one from dad, okay? And then this DNA sequence has to be translated into protein to give rise to basically this is at the end of the day, this is a protein sitting here to give rise to this protein. And the way that actually a cell does this is that with the first allele, they try it, if successful, because one thing here you should notice that is not that all this VDJ will be successful and will be productive and they will give rise to the protein. As I said earlier, if you remember, the protein coding part, it will have a start codon, and these are a triple nucleotide. So you should have a start codon, and then you should have a stop codon, and that should be a multiple of 3. Otherwise, it wouldn't actually translate into protein, and therefore some of this basically wouldn't be functional or productive. And if one allele was unsuccessful on giving rise to a productive VDJ, cell has another chance to try the second allele. And if the second allele wasn't successful, then the cell will die out. And this is the same story, but this time for T cells in a slightly different cartoon. And the reason I actually chose this one is that, so this is one of this, whatever number of regions we have, D gene and J gene, but here, this basically is the insertion that I was talking about. In these regions, some random nucleotide are inserted or deleted, and they basically enhance the diversity of the so-called T cell or B cell repertoire. So, in a sense, so far, I have basically told you that how a T cell or an infected cell process and present a tumor or a mutation and display in the cell surface, and they signal to other T cells that there is some issue here. And from T cells, I've told you that these TCRs, T cell receptors, are very key in recognizing these peptides, as I have illustrated here. So this is the T cell receptor for alpha chain and beta chain. And therefore, again, coming back to the question that was asked, was that to mathematically predict, if we have, firstly, to mathematically predict for a given patient or DNA from a patient, which of these epitopes will be displayed in the cell surface. And if we can predict this, we can actually make personalized vaccine. Another way that, again, it was another question was that, can we make these T cells out of the patient's body? The answer is yes. Basically here, first we have to predict which T cells will have the capacity to recognize patient's mutation And if we can predict that T cells, then our collaborators, our biologists and immunologist collaborators will make these T cells or will train them in the dish in the lab and then inject in the patient's body to do the job. So here, basically, two questions that I just said. Okay, because I'm going to basically tell you about different types of cancer immunotherapy. I'll stop for a minute to see if there is any question here.
Okay, I don't see anything in the chat. Is there any question in the room? If there is, please unmute and ask. But I have a question, not very related, but in a slide that you talk about VDG and you mentioned
VDJ,
that
yes, yeah.
VDJ,
yeah.
and it was very random. If it was not random, the viruses would wipe out the human being from the air, something like that. Is this something that you think happened basically because of the evolution? So we lost those who didn't do it
Absolutely,
randomly.
yes. Yeah, absolutely. Yeah, that is, basically the evolution is the key behind this. Evolution has actually forced not only humans, but other species to develop this. It's very similar, basically, but there are also some differences between different different species, sorry. Yeah, that is exactly it.
Okay, thank you. Any other questions in the room? Well, there is no, so go ahead, please.
Yeah, okay, so and now I'm going to basically tell you about 4 different cancer immunotherapies. The first one is called immune checkpoint blockade treatment, or sometimes I just call it, sorry, it should be here capital, but I missed that. So it should IC either B or ICT normally. So this is immune checkpoint treatment. I'll tell you what it is and how it works. Another one is engineered T cell therapy. This is basically, you will see that we can develop T cells in the lab and put back in patient's body. And this is basically tumor antigen discovery for personalized cancer vaccines. And also another that basically they focus on finding out what is happening in cancer microenvironment, and based on that, they come up with some targeted treatments. Okay, so the first one in theory is actually very simple. So what you see here is a T cell, and this is a tumor cell, and this is the whole story that I described. This tumor cell is actually presenting this antigen or this peptide by this HLA to T cell, and this is T cell receptor sitting here. And in theory, as I said, T cells, as soon as they recognize this, should be activated and kill cancer cell, and they do most of the time. But what happens is that basically in all T cells, there is some proteins that is called PD-1. This is basically T cells break. Okay, that actually prevents T cells to kill normal cells and normal tissues. And for some patients, such as MS patients or those that have autoimmune diseases, most of the time there is something wrong here, and this break doesn't work well, and therefore T cells kill healthy cells. Okay, and on the other side, these tumors, all cells basically have this PD-1 protein. This is another protein, okay? But this protein, most of the time, is switched off. But what cancer cells, again, because of the evolution, what they actually do, they start sending signal, this protein signal, to cancer. And when this interaction occurs, then these T cells, they believe that this is a normal cell, and they forget about killing that. And these tumor cells, basically, they use this mechanism to fool T cells. And what these two scientists actually did, said, okay, what about if we develop an antibody, or we develop basically a protein that can break this interaction? And this is now in the market. It's very commonly used, and this is called immune checkpoint blockade, because they actually block this interaction. Okay, and because of that, they got obviously Nobel Prize for that. Obviously, in theory, it looks very simple, but it comes, with years of investigation and millions of dollars of investigation to figure out how this works and what this antibody or what this protein should be, basically to break this antibody, to break this interaction, basically. And now if we look at the outcome of this or the impact on patients' health, and so this is obviously the proportion of basically survival probability, and it's from obviously you can think of it as a fraction or from 100%. And this first one is obviously chemotherapy, just to illustrate how it works. For chemotherapy, almost 90% of patients, they were actually dying 12 months or 24 months of them. So the survival was almost 24 months in average. Again, it's very variable for person and for cancer. And then this is a slightly different, this is just, here for comparison, is a targeted therapy, but these guys here are different, basically, types of this protein here that can be used either individually or they combine with each other. And what you see here, for example, for this drug here, nearly more than 80% of patients actually, they are alive after 84 months of the treatment or of the diagnosis. And now if you compare this to this chemotherapy, then you will see the difference. And this is now commonly used for skin cancers, for lung cancers, and for cancers that we call them, they are known to give rise to huge number of mutations, because this number of mutations is different between different cancers. And the reason is that basically once basically the number of mutations is high, it's more likely that you can actually find out some T cells that are reactive to these T cells. Sorry, these are reactive to cancer cells, yes.
Yeah, so this PD-1 blockade or anti-PD-1, is it like an oral medication or injection? How does it work?
Yeah, this specific one is either as a form of tablets that actually, oncologists actually prescribe, and taking it should be with very cautious, because it's, as I said here, the existence of this protein in T cells is because it's a brake, because to prevent them to attack healthy cells. So if this drug is not used appropriately, then these T cells, instead of killing cancer cells, they will start killing normal cells, and the patient will die, not because of cancer, but because of T cell attacking to the tissue. Okay, so yes, these are in the market, and surgeons or oncologists, basically, they know which one to give, and most of the time they suggest do one basically or 2 rounds of chemotherapy, and then 1 or 2 a combination, depending on age, depending on sex, things like that, or either individually or combination of them.
Okay, thanks, Hashim. There are 2 questions. Yeah. I try to read the first one. Hamid is asking about, Hamid wants to ask his question at the end, so I go with the Payman. Payman says, in blocking remedy, is there any possibility that blocking happens for the PD-L1 between the healthy cells, or it happens between cancer cells and healthy cells?
That's a very good question. I mean, that is another hot topic that, the aim, obviously, the ultimate aim is that to develop this, and that is actually another challenge here, that developing these antibodies is very tricky. So in a sense, you have to develop these antibodies to only basically break this break for interaction between cancer cells and T cells. Otherwise, it will give rise to autoimmunity. And in fact, one side effect that sometimes we see from these patients is that, for example, we see inflammation in their either intestine or in the gut. Okay, that actually gives rise to very severe diarrhea, or eventually the patient again will die. And one thing that we work is that, is this because perhaps these T cells, they attack normal tissue in the gut and in this intestine or not?
Okay, good, thanks. There is no other question. So we have a question which is for the end, so please go ahead.
Okay, and then again, I think at the beginning there was a question whether we can actually engineer these T cells in the lab, and as I promised, the answer is yes. And what we basically do here, take blood and then extract the T cells. Up to here is basically, these are biologists that they do that, and then we have to basically predict which T cell or which T cell structure, T cell receptor structure. So as I come here, you see that this is a T cell structure or T cell receptor, and this T cell receptor is actually formed here, and therefore we have to engineer this T cells or this TCR to be very specific to patients' mutations. Okay, and once they have it, once they have engineered that, they actually grow them in in the lab. You have an army of them, and then put them back into patient's body. And what will happen is that these T cells actually go anywhere in your body, and they will find these cancer cells, and they will kill them. And this type of immunotherapy is very common for blood cancers and for children. Please don't ask me why, because it's a long story. It will behind that, and we will have to tell quite a lot to find out the principles behind that. But this is more common for blood cancers. But the previous one, immune checkpoint blockade, for solid cancers mainly, skin, gut or intestine, lung, colon, things like that. And then another basically is this personalized vaccines. And this is actually the part that I have already worked, and we have some contribution to this field here. And what we do here, as I have explained a couple of times, we take biopsy from healthy and from tumor, and at the same time we take blood, and by some either machine learning or mathematical modeling, eventually we identify what are those antigens that will sit in the cell surface of cancer patients, and they will activate T cells. And this is really important because this part is very difficult to predict. And once we have that basically then this is called discovering or predicting vaccine targets for that person, or vaccine targets for basically a virus. Once we have that, they make a vaccine in the lab, and then this vaccine is very personalized, because as you see here, we are looking into genome of this person, and then that is actually injected to patient's body. And after that, perhaps some of you might ask that, okay, you told us that cancer cells have some ways of pulling T cells, but what happens this time? So this, if you do this again, T cells will be fooled. But what happens here is that when this vaccine is injected to patient, there are some other cell types. These are called antigen presenting cells. These antigen presenting cells, these are healthy cells. They pick up this from your body or from your blood or wherever this is. And those, because are healthy, their job is that to go and present T cells. And once they have presented T cells become activated, become specific to the tumor, and it's not full anymore. And therefore it goes everywhere in the body and find cancer cells and kill them. And then
may I ask a question?
Absolutely.
The previous slide. So what about after some time? After some time, the same formula of vaccine will work for that patient, or you need a new sample?
That is again, this is a brilliant question. Sometimes, unfortunately, again, because of the evolution, because these cancer cells are under evolution, okay, they evolve all the time. Unfortunately, sometimes they give rise, very similar to COVID. If you think about COVID, it's a similar story. They give rise to a new variant that this vaccine is not actually effective for that. And if that is the case, if that happens, then obviously this vaccine, it wouldn't work anymore. If patient is lucky and we are quick, we can figure out, okay, what new variant was that? And we can actually make the vaccine and help the patient. But most of the time, this new variant that actually occurs is very aggressive, it's very quick, and quickly, it doesn't give us time to do this, one. And the second, these treatments are very expensive. These are not, at the moment, at the society level. Perhaps you haven't seen this, in a normal situation. And therefore, it's difficult for patients to basically pay the cost and ask for a group of experts to do that for them.
I see.
But hopefully in the future, that will be basically part of our, healthcare system.
Sure, thanks.
And another basically factor that makes cancer is a very difficult disease to treat is that, again, because of the evolution, they have developed two other basically characteristics. One is that they actually don't allow T cells into this microenvironment, or if T cells are allowed there, just very few of them, then these T cells, they have been all fooled. In scientific terminology, we tell that these are exhausted T cells. Okay, they don't do anything to these tumors, these cancer cells. And therefore, the question here is that how basically we can develop a way to either restate the functionality of these T cells that are inside the tumor and they have been exhausted, or find a way to basically send these T cells into the microenvironment. And again, there has been great achievement here, and basically it works, and it's a new way of cancer treatment. So is there any question here?
Well, I don't see any question in the chat, but I have a funny question myself. So a T cell will be reproduced by itself, right? 1, 2, 2, 2, 2, 4, something like that, right?
Okay.
Or a full T cell will generate a full T cell or a smart T cell?
Okay. Yeah, that's a good question, and it's actually we need another 2 hours basically.
Oh,
to
okay.
talk
So
about,
don't forget.
No, but I'll try to explain it very quickly and a very simplified version. Basically, T cells and B cells, they originate from bone marrow originally, okay? And then T cells, the reason they actually call T cells because they go to thymus, is a tissue in the body, and in the thymus, T cells are actually checked for 2 things. One, like an army, so you can think of a thymus as an army base that army are trained. The first thing, they want to actually make sure that T cells have receptors good enough to recognize future either cancers or pathogens. If they don't have good receptors, they are killed in the thymus. And then another factor is that, then the question is that, okay, what if the T cell receptor is too good so that they will actually attack healthy tissue? Those again are killed. And therefore, there is a very difficult and challenging question for mathematicians, how to basically find a balance between this so-called sensitivity and specificity of T cell receptors. And this is another basically topic that we work on. And this is really, really complicated. And after that, if T cell goes to this two so-called positive and negative selection, they are allowed to exit thymus and go to blood, and then from blood go to tissue, and in the tissue, some of them become tissue resident, they stay there forever. Some circulate in blood to make sure they have everywhere on the country.
Thank you very much.
Okay, so this is a slight hint on what type of modeling we do. Obviously, it depends to the question. One question we had was that why different cancer patients, when they go under immune checkpoint treatment, we get a different response out of them. Okay, so here we said, okay, can we develop a model to be able to predict immune response? Okay, and this is very important because firstly, from a finance point of view, it's very expensive. And if a patient is not likely to actually respond to that treatment, not only the money has been wasted, but also his or her opportunity has been wasted. So we need to find out in advance that whether that particular treatment will work for that patient or not. So what we said here was that, okay, what about if we take all the data from previous cancer patients that they have gone through treatment, and some of them have responded, some of them have not responded, and then investigate what was behind that. And by that investigation, can we come up with a mathematical model that can help us to predict which patient will be a responder? Okay, and here I don't want to go through the details of this. It will be very boring, but what we argued was very simple. We have one cell gives rise to this tumor. These tumors are actually carrying this cancer mutation, and upon treatment, upon checkpoint treatment, in a sense, we put this tumor in front of some healthy, juicy T cells. And obviously here, T cells start attacking these tumors on the one hand, therefore this tumor is getting smaller, but at the same time, tumors also gives rise to new mutations, and therefore this is dynamic system. And therefore we thought perhaps basically a differential equation model will be appropriate to describe this model. And by doing that, we could actually come up with a model that can tell us. So here I have illustrated the three different scenarios for three different patients. The green patient, after treatment, so here we are looking at relative tumor size. If we normalize the tumor size to 1, very quickly the tumor size goes to below of basically 10%. In other words, 90% reduction in tumor size, and this is a good responder. So this is a good patient to basically go through this treatment. This is a medium effect, and this patient is actually not going to respond because this time, which is a pseudo time, basically is normalized. That tells us that this patient is not going to actually have a tumor reduction of 90% that actually is required for their survival. And then once we basically tested this with some patients that already have been tried, they have actually gone through this treatment, we applied this to 151 patients. We could see that our model can actually separate these two groups. One of them basically are responders and one are not responders. And the importance or significance of this type of models is that this hopefully can help to basically predict which patient will be a responder, which patient wouldn't be a responder, and therefore they need a different type of treatment. This is one contribution, but at the same time, as I said, what I described here, just I used cancer as an example, but it's basically our work is applicable to other sorts of diseases. And actually, from the beginning of pandemic, we started to use our knowledge and contribute to fighting COVID. So this is the very first paper that we published for COVID-19. And basically, once we started this study, and we did it very quickly in my group, the disease still was in China. And we knew that basically that is going to be a threat to the world. And we quickly computationally predicted what part of a virus genome has to be used for vaccine to facilitate basically vaccine development. And the significance of this type of work is that, so imagine you are in a war, and in a war, your resources are very limited. And here, at the time that the disease was in China, there was no sample from patients available for biologists to be able to basically do some experiments. And therefore, it's very important to be able to predict something which is accurate enough to help us to, or help a biologist or experimentalist to go ahead with their experiments. And then after publication, this that has actually has been appreciated in the community quite a lot. It has a good number of citations. Then another basically question, and that was the time that pandemic basically took Europe. We were in the first lockdown. One thing we could see was that we see different outcome from SARS-CoV-2 for different patients. And the very first question that we asked, what is behind this basically variability of disease outcome? And again, please remember that still basically access to samples or to materials was very limited. And that is basically the significance of this type of work. And what we argued was that because there are some other viruses, other families of SARS-CoV-2 virus that we have been living for years, for decades, we thought perhaps in our body, we have some memory T cells from those viruses. And in this study, we basically investigated what is the likelihood of basically immunity, so what we call cross-immunity or cross-reactivity from previous basically families of SARS-CoV-2 virus. And then upon publication this, then still we continue to explore the variability of disease and perhaps variability of response to vaccine that at the time it was just coming out. We discovered another thing. And what we discovered was that, you remember earlier I told you about different people have different HLA molecules. What we discovered here was that actually different patients with different HLAs, they will have a different way of being affected by the virus. So some specific HLAs are more immune to the virus. Some are more likely to, become severely ill. And that was the significance of this disease. And perhaps some of you may hear, in some media or some news that why people with different backgrounds are, getting this disease differently. This is one answer to that. Basically, it goes back to their genome and their basically HLA types. And at the same time, we were basically looking into what models are out there to this and how good they are. And if they are not good enough, then we can actually, or we should develop our own. And another example that perhaps it will make sense. Let me basically get help from Omicron. Okay, so Omicron obviously is not, for now, it's not new, but it's the latest variant of the virus. And actually, it was a threat for vaccination. So at the beginning, we thought, what if vaccines do not work for this variant? And in fact, quickly, it turned out that B-cell immunity to some extent actually do not work, but T-cell immunity is there to some extent, okay? So the question that we ask, why we should always stay, or why we are always behind the virus. We have to stay for virus to give a rise to new variant. And once we see the variant, then we ask the question whether this vaccine is going to work or not. At that time, it's too late. So we ask, what if we come up with a mathematical model and with a computational model that actually put us in front of the virus, and we can actually predict this virus is going to give rise to this variant, and if this variant occurred, if this virus is going to work or the virus is actually not going to work. And that is the way we have to do. And this is something that we haven't published, but here I want to give you two examples. So this is the peptide that is presented in the cell surface from the virus, okay? And then here I have actually put this this bar to reflect the probability of this peptide having T-cell response. If it's reddish, it's very likely that it will have T-cell response. And this is the original, basically, peptide from virus that was used for vaccine development. And as you see, it's very immunogenic. It will have T-cell response. And this is, and then also the same here. And I'll explain what is the difference here. But with mathematical and computational modeling, we basically looked into all the possibilities of new variants in this target. And we basically estimate their probability of being immunogenic. In other words, whether the vaccine will work or not work. If it's red, it will work. If blue is not, it's not going to work for the new variants. And then we cluster them with clustering algorithms. So the point here is that here we have a we have a variant that originally was good. It was used for vaccine development. But if a variant happened here, you see that all of these are blue, meaning that the vaccine is in danger. In a sense, all these variants will have actually chance to escape T-cell immunity. Okay, so in a sense, what I'm going to say is that if the virus gives rise to a new mutation in this part of their genome, then we are in trouble. And now if we look at this one here, so this basically is a peptide that again was used for vaccine development. And with this peptide, again, we have looked at all the possibilities of new variants. We have clustered them. And as you see, more or less, all of them stay red. And the message, the scientific message of this is that actually, this is a very immune or very secure peptide to use for new generation of vaccines, because whatever mutant or variant virus actually forces here, always we will have T-cell response. But this one is not the case. So in a sense, what we are telling to the community is that for the next generation of vaccines, what you have to do, basically you have to avoid making vaccines from these guys, because this will be very unsecure, unimmune, but this one will be very secure. Okay, and if we basically we are in the final version of wrapping up this, upon publication, which actually it will require a number of experts to review the work. If published, then it will be accessible to other scientists and to basically decide about future vaccine development. And very last couple of slides, just to tell you that this cancer or vaccine is not the only basically contribution that we have made. As I said, because we need to understand how immune system works. Basically, one place in the body that gives rise to 70% of immune cells is gut and intestine. And the reason is that in gut and intestine, there are billions of different microbes and viruses, and T cells actually go there to be specific to that microbes and those bacteria. And therefore, it's a very good tissue to understand how T cells and B cells interact and signal and develop in the gut. And these are very key publications that we have made into either tissue development or immune development in the gut. We have also looked into how adaptive immune system goes down as we age. So you have noticed that once we are aged, we are basically susceptible to different infectious diseases. Vaccines do not work, and we become ill more often. And in this, actually, we identified which basically family of genes actually go down in age. And this actually, again, would help to develop some treatments or some new generation of vaccination for elderly people. Yeah, and I think that's it. And I'll stop here. It took too long, but hopefully, I was able to make some points, and I'll be happy to take further questions.
Thank you, Asham. Thank you very much. Very well done. Very informative. Great presentation.
Thank you.
We have 3 questions in the chat so far. The first one is it really true that mRNA vaccines are better than the other types?
Well, it's difficult for me to answer that. I wouldn't be able to tell whether they are better or not. But what I can tell you is that, as you noticed earlier, that we have DNA, we have RNA, then we have basically protein. And therefore, so far, most of the proteins were built of proteins. Proteins are built in the lab, and they put it back to the patients, and then, all the story. But questions, or scientists like Pfizer scientists, they said, okay, what if we actually go one step back and make vaccines from RNA and leave it for body itself to translate the mRNA to protein? And this is a better system. Obviously, in theory, it's a better system because part of the job is done by the body, but also it has some other complications and difficulties that I'm not expert in explaining.
Okay, good. Thank you very much. So the next question is, Payman says, has any study performed yet regarding the cancer cells to know how they mutate into new forms and grow? So that knowing how to make a difficult flaw in their chain of growing. So technically he wants to know, as we know how the cancer cells are growing, is there a way that we can cut their way and, kill them at source?
That's a great question. I mean, there is actually a massive field that they study cancer evolution and cancer development. It's not actually, in my area of expertise. But they do that for each basically cancer, either at cancer type level or even at the patient level. They have defined what they call the signature, the basically mutation signature of that particular cancer. It's very complicated, as you can imagine, because all the time the cancer is under evolution. Different factors actually, different forces are applied to cancer to give, rise to new variants. Yes, it is. Yeah, there are massive amount of publications, tons of them. Yes.
Okay, good. So the last question on the chat is, you said that in the cost of a couple of these treatments are so high that public don't have access to it. And you mentioned that hopefully one day that will be a part of the public health. Do you know probably when? Is there a light at the end of the tunnel? Is 5 years from now, 10 years from now? What do you think?
Well, again, great point. Obviously, my hope is the sooner the better. I don't know the answer, but what I can say is that there are different studies, trial studies. Actually, we have just started one. We managed to secure money for basically brain cancer. And with that, basically, what we first noticed was that we discovered basically a way that cancer silence T cells or fools T cells. Okay, it's a different basically way from what I described here. We published this. Publication came out actually a few months ago, and then by that observation, we asked one funding body to give us money to basically hire about 50 patients, to register 50 patients. And we test with this basically new treatment in these 50 patients. Obviously, first it has been basically tested on mice. Okay, so that phase has been passed. So the next is a cohort of patients. In our case, we have budget for 50 patients. Point being is that, yes, these are not commonly used in healthcare systems, but every now and then we see some basically trial studies that, depending on their money, they register either 100 patients or 1,000 patients or 20 patients, and they offer this. There is some good things and some bad things about that. Good is that basically most of the time, these basically treatments have passed the previous phases, and it's likely that they will actually work. But that is that it's not granted. Basically, you have to basically throw a coin. It's 50-50 from cancer patient point of view that it will work or not. But if it will work, they will actually increase their survival quite a lot. Usually, they go ahead with that.
Okay, good. Thank you very much. There was a question which was asked before but postponed for the end. It says, based on what you explained about math model and training sets and different probabilities, I believe the prediction was non-linear. If you could disclose how many parameters did you predict in your model?
Yeah, I don't know which one the question is referring, because we have done actually different modeling. For instance, obviously this is one, this is another one, and here we had not actually many parameters. It was just a few parameters that we played with. Yeah.
Okay, he says regarding, well, Payman had some questions, and well, Hamid, I think you have to unmute and ask the question yourself.
Okay, sorry. Yeah, so you guys discuss about different probabilities, what Payman asked, like in one of your slides. If I look into the chat,
you mean the probability of the DNA mutation and
exactly
from T to G and different amino acid?
Yeah, but in general, my question is about the mathematical model, because what we do right now in engineering, most of our models are linear. We cannot predict many parameters. We don't have that much of a strong model. I just want to know that based on what you created over there, is it nonlinear? And if it is nonlinear, how many parameters you predict for your cases based on the data that you have and based on what you train for using artificial intelligence or using any statistical method?
Yeah, there are a few good actually points. There are different steps, different stages, and obviously we do not do and we cannot do all the stages ourselves. So let me go back basically to here. As I said, from the time that basically cancer or a healthy DNA encounters a mutation, that is the beginning, okay? And then this stage is comprehensively studied with those expertise in cancer evolution and cancer signatures. Okay, they have quite a lot of good publications all the time comes out. Most of them in the UK from Sanger Institute, because there are a number of leading scientists there. And then from this part up to here, basically, that is something that we very generally we call it peptide processing and presentation. And here again, there are the dominant model here are deep neural network models, and they start with obviously a DNA sequence or protein sequence, and different models all the way to predict which ones will be sitting here. Okay, and this part is kind of mature, and the models that are out there are kind of accurate enough. And for that reason, we didn't spend on this part here, but rather what we have been focusing, and it's more complicated, and for that reason has not been addressed, is that from those that are sitting in cell surface, that are again thousands or tens of thousands of them, which ones are more likely to activate a T cell? In a sense, which one basically will make this interaction? Okay, in a sense, we won't actually predict. Here also I have put actually a circle, but here I refer to a short peptide, short protein sequences. And again, here there are different models. We have taken, for example, hidden Markov model. We have taken Bayesian, approaches. We have developed. Actually, my PhD student, is just submitting a paper that she has developed a deep neural network model that goes through all the previous data that are available, those that were immunogenic, meaning that they were actually capable of activating T cells, and those that weren't. And with that model, now she claims that she can fairly accurately predict which peptides are immunogenic, or in a sense, we can predict targets for vaccines, basically. Yeah, well, you are right. I mean, most of the time, it's very complicated. Different models are applied. In some very limited situations, some linear models might work. Most of the time, there are some more generalized linear models or nonlinear models.
Oh, great. Thank you very much. Okay, so I don't have any other questions in the chat. Is there any question in the room?
No? May I ask a general question?
Yes.
Dr. Kouhi, thank you very much for a great presentation. I've really enjoyed. I learned a lot.
I'm happy to hear that.
Do you hear me or not?
Yes.
Yeah, absolutely. Yes. Yeah.
Okay. Based on your study and experiments, what types of cancer are these days easy to cure? Prostate cancer, breast cancer, or which are hardest and which is easiest, based on your general experience?
No, actually, it's a good question, and it's a very hot topic. And that is one reason that we have different types of cancer immunotherapies, okay? For the easiest to treat, I mean, it's because this basically all recorded. Perhaps some of my colleagues, expert colleagues, wouldn't actually like the answer I gave here. But basically, from immunotherapy point of view, not all of them, some immunotherapy point of view, skin cancer is basically commonly used, or patients with skin cancers are have chance to get in particular this treatment. And actually in United States and in some countries, these have been actually approved and are actually applied, okay, this treatment. And that actually works for skin cancer, for some sort of lung cancers, for colon cancer, or in a sense, those cancers that gives rise to a huge number of mutations. For some other cancers, such as blood, this is not going to work. Again, if you ask me why, that is the reason we are actually get paid to investigate what is the reason. We don't fully understand. On the other hand, there are some other perhaps physical factors that actually play a role. For example, if a breast cancer is diagnosed in very early stage 1 and 2, which is still a primary tumor, then obviously the surgery is the best option, and quickly they will actually took off the tumor or the tissue, and then you are free. Basically, you're free of cancer, and that actually is very less likely to come back, unless there are some cells that has actually gone through other part of your body. Yeah, so hopefully this answers your question.
Yeah, thank you very much.
Okay, so I don't have any other question in the chat. Any other questions in the room? If yes, please unmute and ask. Okay. اوکی، به نظر میاد که ما دیگه سوال دیگه نداریم، میتونیم ما دیگه فارسی با هم خوش بش کنیم. خسته نباشید آقای دکتر، خیلی ممنون، دستتون هم درد نکنه، خیلی بیشتر از زمانی که من ازتون قول گرفته بودم برای ما وقت گذاشتید، ممنونم. اگر فرمایشی دارید برای ختم جلسه های دکتر بفرمایید که با سخن شما ختم جلسه رو اعلام کنیم.
خیلی متشکرم. من هم از شما هم اونایی که ارگانایز کردن این رو و از همه دوستانی که اینجا وقتشون رو گذاشتن و گوش کردن و برای من اکچولی خیلی جالب بود. کوئسچن ها همشون کوئسچن هایی بودن که part of our research و آره اول که وقتی شما از من پرسیدید و من خورده مردد بودم که به اصطلاح بیام یا نیام، خورده نگران بودم که نکنه بیام یه سری چیزایی بگم که بورینگ باشه برای دیگران، ولی at least from questions point of view نظر می اومد که اصلا
من یواشکی بهشون گفته بودم موضوع چیه، سوالا رو آماده کردم آقای دکتر.
نه به هر حال ما افتخار بود اینجا و حکم میکنم که اصلا یه خورده چیزی با هم مثلا یاد گرفته باشیم با هم دیگه اینجا.
آقای دکتر بنده هم ضمن تشکر و سپاس از سخنان بسیار عالیتون، مستمعینتون آدمای فهمیده بودن همه.
absolutely.
این مطلبایی که فرمودید خیلی های کلاس و ریزبینی بود. بنده به نوبه خود سپاسگزارم از بهشاد عزیز، جناب آقای محقق ریاست محترم کانون. واقعا یکی از برنامه‌های بسیار عالی و مفید برای همه است، مخصوصا برای پیرمرده مثل بنده. سپاسگزارم از لطفتون.
موهای سفید هم قایم کنم.
خداحافظ میکنم. آقای محقق بفرمایید.
از طرف هیئت مدیره کانون مهندسم از طرف شما صمیمانه تشکر کنم، قدردانی کنم و این قول رو از شما بگیرم که در فرصت‌های دیگه این شانس رو به ما بدید و با تاپیک‌های دیگر و با نتایج ریسرچتون ما رو خوشحال کنید و جامعه مهندسین ایرانی کانادا و سایر گروه‌هایی که حضور پیدا می‌کنند در این جلسه از دانش و فرزانگی شما بهره ببرند.
خواهش میکنم. ما افتخار بود و حتما اگر چیزی باشه که بتونیم اصطلاح شیر کنیم why not و آره basically آره قسمتی از کارمونه. actually ما وقتی که یه ریسرچ میخوایم انجام بدیم، خیلی از شماها این مسیر رو خودتون رفتین میدونین، حالا شاید یه خورده depending on کشورهای مختلف خورده مختلف باشه. یونیورسیتی به ما خیلی پول نمیده، باید مثلا یه پروپوزال بنویسیم، بعد اینو بفرستیم ارگانیزیشن ها بره through reviewing و به ما پول بده. و part of that is basically public engagement and outreaching. بنابراین علاوه بر این که حالا من خیلی خودم دوست داشتم اینجا یک اصطلاح گروه دوستانه و چیز بود که میتونستیم با هم رد و بدل کنیم اینا رو، این به ما هلپ میکنه که به فیوچر باز بیشتر به اصطلاح باکس ها رو تیک کنیم و بگیم که آره ما این اکتیویتی ها رو هم داریم و لطفاً به ما بیشتر فاند بدیم که کانتینیو کنیم این ریسرچمون رو. no منت اون سر کسی.
مرسی، ممنون. آقای محقق اگه فرمایشی ندارید من ختم جلسه رو اعلام کنم.
تشکر میکنیم از شما و همه عزیزانی که در جلسه شرکت کردن، با سوالهای خوب، با حضور خودشون ما رو خوشحال کردن. سپاس و قدردانی مجدد از همه شما.
متشکرم. بعد از ظهر خوب و آخر هفته خوبی داشته باشید. خدا نگهدارتون.
متشکرم همچنین برای شما. خدا نگهدار.
خدا نگهدارتون.